So we'll talk about some of these periods, what happens during them, and I'm going to just let you know what I expect you to know, because there are a lot of changes that occur in each one of these stages, particularly the embryo and the fetus. The zygote is more of a traveling kind of a period. So let's talk about some of this. So of course, conception, I think we all know how it happens. We may even practice it from time to time. And again, the ova is going to be released from the woman's, from her ovary, of course, and it alternates every month, left or right. So there are two ovaries, and each one will be producing an egg on every month, but alternating left and right, which ovary actually releases the egg. Sometimes, of course, you have 2 eggs released, or 3 eggs, depending on, again, the woman's fertility, and that's how, of course, you have multiple births. People taking fertility drugs. This would, of course, cause that to be more likely. So the ova is going to travel from the ovary to the fallopian tube, where it awaits a sperm. Now, this is not your intention. This is the egg's intention. And the egg, it's all about the egg. It's not really about what you want, right? So we try to prevent this from happening through, of course, various methods. But for the most part, the penetration by one sperm, who is the successor. And I look at this as kind of the guy who travels through and makes it upstream, because I mean, it really is quite of a feat to be that one sperm that actually penetrates the egg. Now, I've got here penetration by 1 of 300 to 500 sperm. I need to qualify that. In a teaspoon of seminal fluid, you're looking at millions, maybe 300 to 500 million sperm that are going to be vying for a chance to be that one. It's only 300 to 500 that make it this far. The rest of them, of course, are not successful. So this is only a few when you think of 300 to 500 million that actually are released. So one of these 300 to 500 that are eligible swimmers is going to be the one who is going to impregnate the ova. And I liken it to, you always think of these things in your mind to help you learn. I used to think of that one sperm as the swimmer, right? The fast swimmer going upstream. It's almost like a salmon spawning, going against the tide. And he's got like this little flag, and he's going to be the one to like put his flag in the mountain. I'm the one that made it to the summit, right? I am the winner. In any event, maybe not. This is again how I look at this is one particular sperm, and it cannot be impregnated by more than one sperm. Again, that's not twins. I mean, you could have 2 eggs, of course, but one sperm can only penetrate 1 egg. There are some proteins that are released to prevent the impregnation of 2 sperm on the 1 egg. So again, twins would be 2 sperm, 2 eggs, or 1 egg dividing in 2. All right. Once that happens, this single cell, which of course is the exchange of DNA, which we talked about last class, the 23 chromosomes from each one of these gametes, will result in the full set, and that, of course, will be a single cell now called the zygote, which will then begin a journey to the resting place, which will be the uterus, where that child will stay for the next 9 months. So here is the period of the zygote, and again, weeks 1 to 2, after fertilization or conception occurs, the zygote is going to travel down the fallopian tube and eventually become implanted in the uterine wall. So during this stage, things can happen. Nothing, we'll talk about teratogens, but the zygote is relatively impervious to teratogens. So if you're taking a drug, or if you're taking alcohol or some other kind of chemical, or something happens toxin-wise, this is not going to affect the developing zygote. What can happen, of course, is a fallopian tube pregnancy, where the egg, or the zygote in this case, gets stuck, if you will, in the fallopian tube and doesn't travel all the way down to the uterine wall or into the uterus. And sometimes these can actually result in a uterine pregnancy, which can be very, very dangerous and very painful. I've heard of some women, probably not so much now because they don't use it as a form of contraception, but there used to be a device called an IUD, or an intrauterine uterine device, and it would sometimes become dislodged in the woman's body, and it would allow her, of course, to become pregnant because it wasn't sitting correctly in the uterus. And the woman could become pregnant with the IUD still in. and you can't remove it once it reaches a certain period in the pregnancy. So I knew this one woman who actually had an intrauterine pregnancy with the IUD in her, and she had to go through the pregnancy, and the IUD actually was shifted and it was causing a lot of pain. Her baby was born roughly about 6-7 months into the pregnancy, so very, very, very early, and the IUD was actually pressing against the baby's head. They could see this on an ultrasound. So of course, cesarean section, and they were worried that the child would have, brain defects, or would be, of course, compromised during the delivery. But everything turned out okay. Child is now in his 20s and totally normal and has a child of his own. So everything seemed to be okay, but that was very, very lucky. So tell me why the zygote is impervious to teratogens. Once you start getting into this stage and on, 3 weeks and on, this is very, very, very critical. Anything that happens during this period is probably the most sensitive time and the most dangerous time for the developing child. But here, there is nothing that can happen due to teratogens. Why not? Yeah. That's worse. She doesn't know she's pregnant at this time, so she could be taking a lot of drugs. So, but why would the effects of that not affect the child? So if you don't even know you're pregnant and you're drinking and taking heroin, it's not going to affect the zygote. Yeah. Yeah. The zygote is not implanted, which means what can't happen? There's no exchange of bodily fluids. There's no blood being circulated through the child at this point in time because it's still going down the fallopian tube. So it is not getting any of the bodily fluids from the mother. So things can happen here, and if they did, most likely nature would take care of it and the child would spontaneously abort. All right? And the mother wouldn't even know it ever happened. You wouldn't even know. Down, we get into the period of the embryo, weeks 3 to 8, and this is, again, a whole different story. Once this happens, I'm going to show you the actual implantation and what marks this particular stage. Once that zygote is implanted in the uterine wall, and it says weeks 1 to 2, it can happen. I mean, this is a range. Usually it takes about 6 to 7 days for the zygote to be implanted, but they say 1 to 2 weeks, so I mean, it can vary. So roughly the first 2 weeks is this traveling and then becoming implanted in the cervical wall or in the uterine wall. Once that implantation occurs, then we reach the period of this embryo. And that this is the point when the woman will stop having her period. So her menstrual cycle will cease once that is implanted in the uterine wall. She still may not know she's pregnant. And again, tests today are pretty sensitive now. They can actually test for hormones that can occur immediately after conception. But these are, again, very, very, they're not the norm right now, but they are available. And of course, now you don't have to wait until the rabbit dies, right? We have all kinds of tests that you can perform that will test for these levels in your blood to determine whether you're pregnant or not, or in the urine. So this is going to be a lot of stuff going on weeks 3 to 8, and this is really going to be important for brain development. This is why this period is so critical for anyone who is taking any kind of drug or alcohol, or doesn't matter whether it's a street drug, prescription drugs as well. And the unfortunate side of this is if you're taking drugs for, some women take drugs, hormones, sex hormones, to help them, if they have, let's say, fertility problems, they take a sex hormone to prevent miscarriage, but that can actually have an effect on the developing child. Or if you're diabetic, or if you have, if you're a bleeder, you're taking anticoagulants. Anybody who has epileptic seizures, you're taking anti-seizure medication. Aspirin, caffeine, coffee, drink of wine. All these things can have effects on the developing child. And it is pretty much here that you have a lot of the emphasis. Not because it can't affect you later on in week 9 to 38, it can. But this is when the brain is developing and the central nervous system. So anything that's going to damage you is going to be really, really profound at this point in time. Because again, the internal organs, the 3 layers of this particular layer here, and I'll show you this. This is the actual embryonic tube. And the embryonic tube is basically the development of the brain. So this tube looks like a salamander, and I'll show you a picture of it. It closes in on itself. And when it folds in, it has three specific layers. And again, this is outer, middle, and inside. So named ectoderm, meaning the outer layer, mesoderm, the middle layer, and endoderm, the inside layer. And the ectoderm is the outside layer. So we're going to find things there like skin, circulatory system, the peripheral kind of nervous system, hair, right? So anything on the outside. The mesoderm, more in the middle layer, is going to be things like muscles, bone, circulatory system. And then the inner layer, the endoderm, is going to be things like the lungs, some of the organs, right? So deeper within. But this whole thing, again, is going to be, have a top and bottom. The top is going to be the brain, and the bottom, of course, will be the spinal cord. So this is a really important time for the developing CNS and, well, of course, the brain and spinal cord and the peripheral nervous system as well. This is also the period when the amniotic sac is going to fill with fluid, and the umbilical cord is going to connect the embryo to the placenta, which is where the child is going to be nourished. And of course, anything toxin or nutrition as well, anything that the mother ingests is going to cross the placenta through the umbilical cord to the developing child. So basically what goes into her body is going into the child's body as well, good or bad. And the sad part is there is a membrane in between the mother's blood, and they don't connect. The mother's blood and the child's blood don't mix, but there is a membrane that allows nutrients to pass through, and unfortunately, it allows toxins to pass through. You'd think that it'd be more like the blood-brain barrier, where there would be this kind of shield against things that are molecularly toxic, but it doesn't. It allows these things to pass through as well. So this will become clear as I go through this. So here's that little trip I was talking about of the zygote. So here's the ovary, and again, it alternates. Ovulation, the egg will be released. Fertilization can occur within 12 to 24 hours. The sperm can live roughly for 2 to 3 days as it makes that journey, so that can also happen during that time period. It starts to double every 12 hours. These cells start to double, and then after a while, of course, it becomes very, very big. And not big, but it becomes very big considering you're only one cell back here. As it travels down the fallopian tube, then implantation occurs about 6 days. So again, once it implants into the uterine wall, this is what's going to form the placenta right here. Is this cell adhering to the uterine lining. And that is going to become, of course, the placenta. So this, again, is the first 2 weeks of pregnancy. And, of course, woman has no idea at this point. Here it is, the implanted zygote. Now, this is not the actual size. So don't think, oh, my God, my first baby picture, aren't I cute? This is, again, with an electron microscope, millions of times enlarged. You wouldn't even be able to see this. It would be the size of a dot at the end of a sentence, right? So this is, again, the uterine wall. This is the zygote attaching to the uterine wall. And at this point is when she will stop having her menstrual cycle. Inside is called the germ disc. And this is what develops into the infant. This is not the baby right here. The baby, of course, is inside in this, what we call the germ disc. And that will start to develop into the child. Just to show you how fast this happens, it's absolutely incredible how quickly this development occurs. So down here we have, this is in weeks, week 3, and again, I mean, this is what they have here is an inch. This is kind of off scale. I think it's the biggest inch I've ever seen. But nonetheless, this would be roughly, the size of a, seed in a piece of fruit. It's a tiny, tiny, tiny little thing. And then as you can see, even 1 week later, look at the difference in 1 week later. It's not double. It's like, huge amounts of change. Just 3 weeks later, look at this. Now we're starting to see even human features. The eyes, of course, are kind of like, they're almost extraterrestrial looking. They are on the side, almost like compound eyes, like a fly. That will go away. So all these things start to take on much more human proportions and quite top heavy. Because the brain is developing during this period, it's a very rapid period. So the head becomes about 50% of what the child is. So it's not a pretty picture, all right? But then again, this is again how development occurs. The arms and the legs will start to develop, they look like little flippers. So again, in time, but again, it's only 8 weeks, right? And just in 2 months, we have here, if you look at this, you could say, yeah, this is a human, you know, hopefully. But you can tell that it will eventually be a human and not something else. This is actual pictures here. Here we got 40 days, 45 days, 2 months, and look at this. 2 months, you see an eye, you see the actual shape, you can see the organs starting to develop. 2 months later, I mean, look at the mass difference here. We've got the vascular system in the brain, we've got the arms, we've got the legs, the ribs, the skeletal system is formed, umbilical cord here, all of this in just 4 months. So it's going from here to here is not all that much of a time frame, but it's massive what goes on. And of course, the child is immersed in the amniotic fluid, which acts as a cushion to protect the child from any kind of force or a blow. If the mother were to fall down, this would protect. So they're kind of buoyed in this water. It also maintains temperature because the child is unable to maintain their own temperature, which is one of the problems if a child is born too early, is that they cannot maintain their own body temperature or digest food, of course. So this maintains a constant environment and helps the child to receive nourishment, to eliminate waste. The child will actually defecate and urine in this area. So by the time 9 months is up, this particular amniotic sac is filled with, yes, you guessed it, urine. So this is how you spend most of your time swimming around in this. But I mean, it is eliminated to the mother's waste. It's not all like, it's not like going into a public pool. Sometimes that's worse. But nonetheless, it's not this fresh water that you'd think, some alpine springs. It can be, I mean, that's the truth, right? So in any event, we all survive it, and after a while, the child will swim through it, and not for long, because of course they get bigger. But for a while, they'll start to move around in there, and the mother can feel it by about here. Even 3 months, some women can feel the child actually moving around inside the placenta, or sorry, in the amniotic sac. This is, again, the events that occur during this particular embryonic period. Now, look at all the stuff that happens. I'm just going to go through it just so you get an idea of how dramatic this is. But I am not going to ask you on a test. Week 5, I had a prof that did this. I took developmental psychology at St. FX. And every week, not every month, every week, we had to know what happened during each one of these weeks. That, to me, is not knowledge. That is just pure torture. And I think it's ridiculous. I think it's more to get a bigger understanding of what the period, what the actual, stage encompasses, not each one of these weeks. And let's face it, not all this is going to happen identically for every child. These are, again, averages or norms. It may not happen until week 4, week 5, some of these things. So I just want you to get an idea of how dramatic these changes are during these particular periods. So the main periods of the embryonic period, again, those 3 layers, which is important, ectoderm, mesoderm, and endoderm, that's going to occur. And again, the embryo is curved and it folds in on itself. And as I mentioned before, the outer is going to form into the neural tube. The neural tube is that part of the, well, it is a child, but again, most of this will be the brain and the spinal cord. Inside, again, the heart will form and begin to beat at 4 weeks. Can you believe it? Endoderm, again, lungs, all this stuff. So I'm more interested here in these 3 particular layers emerging during this period, not the week, but during the embryonic period. Moving on, the ears, mouth, throat, arms and legs, hand plate, the heart divides into the chambers. And again, the brain differentiates into the different sections, forebrain, midbrain, hindbrain. And most of the development, of course, is going to occur during this brain formation. Here, almost 1 inch long, fingers emerge, primitive facial features, sexual differentiation. Now, you can't see the sex at this point in time. It's starting to differentiate. You won't be able to see that until the end of month 3, month 4, where the actual genitalia for males, you can actually see it on an ultrasound. But at this point, you can't see it, but it is happening. Structures and organs are present. Ovaries, intestines are evident. And then again, assumes a more human appearance. So these are the major parts of the embryonic period. A lot of stuff going on, right? A lot of stuff. Here's the neural tube I was telling you about. And this is, again, the neural plate up here. It folds in on itself, as you can see. So this part here is flattened out. This then will fold in and form this kind of a tubular structure. Up here is the future brain, and down here is the spinal cord. So a couple of things can happen here with improper development. Number one, this has to close over, or the fetus will actually abort, or the embryo will abort at this time spontaneously. Some children have been born with this open, but they do not survive. It's called anencephaly, which translates to literally lack of head. So you can't live without a head or a brain. So this would not live. Down here, however, the posterior end, if this fails to close, the child can live. They can now surgically rectify this. There's now a fabulous branch of medicine where they can actually go in and operate on developing fetuses, which just blows my mind. They perform surgery. They go into the amniotic sac, into the mother's abdomen or through the cervix, and into the child, and actually operate and fix some of these abnormalities. One of them would be to fix this problem right here, which is spina bifida. So if this does not close, this is spina bifida, and easily rectified through the mother, or preventative, I should say, by eating folic acid. So that will prevent this from happening. But that's exactly how it happens, and now, of course, they can go in and help that to close if it doesn't close on its own. So here, the neural plate, this is an interesting aspect as well. Your book, I don't think, talks about this too much, but this is, again, the area or the time when the brain is developing. And during this period, what we have here, because the brain is developing, in this neural plate we have these glial cells. And glial cells are kind of like regulator, they're almost like regulator genes that are going to migrate out in sheets. I think there's about 6 layers of these migration. And these glial cells will migrate to certain parts, and they will rest in that certain area. Then another branch will start to migrate. This is called neurogenesis, or neuronal migration. And that just means a developing brain, all right? So as these cells are migrating out into different areas, they form structures. And if a group of cells lands in, let's say, an area over here, then they take on the function of the surrounding cells, just the property of how these genes work or how these cells work. So surrounding neurons will act like surrounding neurons, and that forms groups or functions. This is what gives rise to the various structures of the brain. So because they are migrating out, these neurons can basically be anything that they want them to be at this point. They haven't really differentiated into any particular structure. These are stem cells. So if you can get the stem cells at this point in time, it is optimal, because this means that they can become anything you want them to be. So for example, let's say in Huntington's disease, where the person suffers from an inability to produce dopamine in the substantia nigra, which is an area in the basal ganglia. Substantia nigra is important for developing or producing dopamine, which is involved in motor fluidity. And people in Parkinson's, I should say Parkinson's, have low levels of dopamine, which causes them, of course, to have the disease at the onset of around 30 to 40 years old or 50 years old. So if you could get stem cells that are undifferentiated, they haven't really become a particular structure at this point in time, and harvest them and implant them into a person who has Parkinson's in the area of the substantia nigra, surrounding cells will take on that particular property and become dopamine-producing cells. And that's the whole idea around stem cell research. Again, it's controversial because they're getting these cells from aborted fetuses. But at the same time, there are, here's the question. And again, there's no right or wrong answer. But if women are, donating their eggs or freezing their eggs, and let's say they have, 50 of them being frozen, and they're going to take them when they need them for when they decide to have a child. And now they've reached a point where they don't want any more children. You still have these eggs that are viable. What are you going to do with them? Does it not make sense to use them for medical purposes to save people's life, or for research for treatments for these kinds of diseases? Or do you just discard them or use them for experimental investigation, right? So this is where bioethics comes into play, which is biology and ethics trying to come up with some way to, answer these questions that arise. Nonetheless, it's still happening, and it's going to happen. We can't stop it. So this is, again, what's happening here in the neural plate is this what we call neurogenesis or corticogenesis, which is the beginning of the brain or the cortex. So clearly you can see if anything goes wrong in this period, the child can suffer dramatic effects, central nervous system problems, learning and memory, seizures, all kinds of mental challenge, and I mean severe mental challenge. So again, all about the central nervous system and the actual cognitive functioning of the child will be affected. So this is neurulation, and this is again formation and closure of that neural tube. And it occurs about the 25th day of gestation, so not very long into the pregnancy. Again, the posterior end, if it does not close properly, is spina bifida, and the anterior is anencephaly, which of course would not survive, right? And probably wouldn't even, it would probably be spontaneously aborted because nature takes care of severe deformities like that. Any other kind of migration problems, and again, we're talking about this area here where the neural plate is forming and these cells stem cells are migrating out to become, future brain structures. If there are problems in corticogenesis or neurogenesis, all the same thing, the migration process can produce some strange results. One of them, of course, could be problems with the corpus callosum. And the corpus callosum, of course, is that nerve tract of fibers that connects the left and the right hemispheres, right? Some children are born acolossal. They have no corpus callosum, in which case they may live their world like having two independent brains. So the left brain and the right brain, and the functions that go with each of those two halves don't communicate the way they do in most people. Now, it's not like walking through the world with only having, the left and the right separated, because you have the visual system, and the visual system will allow both hemispheres into, what's going on. But there are some things that are very subtle that people will tell you happens when they are born acolossal. There has been an operation as well where people can have their corpus callosum cut. It's called split brain. And oftentimes it's cut because of epileptic seizures. And this prevents the severity. And people who have split brain see the world basically in two independent views as well. So their left brain processes detail, their right brain processes, the whole gestalt. And sometimes these people report that on some occasions they have gone to do something with their left hand, and their right hand will come and pull it away, right? It's almost like you have two independent modes of thought. So there are some strange things that can occur with the corpus callosum. Or if there's migratory problems with the ability to transfer information from one hemisphere to the other, it could result in attentional problems. There have been some links to people who have severe forms of ADHD with problems with inhibition of the left and the right hemisphere. The right hemisphere is usually involved in attention, right? So if you have problems with the transfer of information from, let's say, one hemisphere to the other, one hemisphere may not be able to inhibit the right hemisphere, and vice versa, which means you have attentional problems, right? You can't inhibit the attentional aspect of that. So there are a multitude of things that can occur, problems with the corpus callosum. Another one will be subcortical, and that again is below the level of the cortex. So it could be motor dysfunction or other issues to do with things more in the midbrain. And again, these can all occur due to migration. There's one particular problem, I don't expect you to know this, where there have been some research that shows the development of what they call ectopias. Ever hear of ectopia? Ectopias? Ectopias are what, in layman's language, they call brain warts. And they're displaced neurons. So they are neurons that don't follow the proper migratory path. They become transient, they become dislodged, and they will rest in an area where they shouldn't be. So let's say you have a few of these neurons, or let's say hundreds of these neurons, that don't migrate to where they should go. And if they settle in an area, let's say in the auditory cortex, that they believe may give rise to some issues with dyslexia, people who are unable to hear the phonological aspects of sound in human speech, and that prevents them from hearing the subtle nuances in very, very small the phonemic structure of language. So these are some of the research things that have been going on to try and find out what happens during migration of neurons. So a very important time in the child's brain functioning or development. So here we have a 3-week-old embryo, and this is what I was saying, it looked like a salamander almost. And this is, again, the top of the tube, and this is the bottom of the tube. And again, this should close. If it doesn't, it's spina bifida. And again, we can actually surgically fix this. Over here, 5 weeks later, and look at this. And there we are, and there's no question that this is, you know, an embryo. Over here, you might not know what it is, but here, there's no question. Yeah? Well, if that doesn't close, it doesn't matter when it happens. It has to close. So if it doesn't close, then it's spina bifida. And usually that is going to happen around that 3rd week, right? So the time period would be about the 3rd week. But it has to close, or else it will be spina bifida. Okay? But again, there's ways to prevent this. Diet, folic acid, surgery, right? This is showing the neural tube closure. So up here is successfully closed, as you can see here. Over here, and again, that's why it affects the spinal column, because it's in the back end of the developing embryo. And here it is here open. So starting to close right here, right? So again, this is what it really looks like. It looks like, what does it look like? It looks like a little guinea pig to me. I'd be worried if I saw my picture, but in any event, this is a real slide of the developing embryo. So then we go into the 3rd period, which is the fetus. And around week 9, again, the ovaries and testes start to be differentiated. Week 12, circulation. Week 16, which is about 4 months, movement can be felt by the mother. And age 23, we call the age of viability. This, again, is not set in stone. This will have a window, I guess you can call it a window of opportunity, where a child who is 23 weeks, it's viable, meaning that they could possibly survive if they were born this early. It doesn't mean that a child who was born at week 22 or even week 21 couldn't survive, but most likely have a difficult time. So week 23 is just that, you know, optimal period that they say the child could have a higher survival rate. So I'm just going to show you here, and again, it says weeks 23 to 25, right? So again, depending on the actual child, weeks 23 to 25 could be, you know, a chance where they could, that this early fetus could survive. If I were to ask this on an exam, I'm not going to be mean and say, okay, the age of viability is A, week 23, B, week 24, C, week 25, D, all of the above, right? I would not have it that ambiguous. I would ask you A, week 10, you know, B, week 24, C, week 30, right? So I'd make it more within range of the actual weeks, okay? If that were a question on the midterm, that is. I'm not saying it is. So again, here are some of the things that are happening during the fetal period. So again, I'm not going to have you memorize, which is what it would be, each one of these weeks. But I would think that this is an important aspect, the age of viability. So again, the head here is about 50% the size of the fetus. Now it's going to start to develop more in the bottom end. Fetus can now open and close its mouth, turn its head. Here we have fingers and toes, arms and legs starting to kick, breathing, reflexes. Heartbeat is now audible with a stethoscope. Fingernails, hair, brain development is actually pretty good now. I mean, this is almost done. This will continue, however, but well into after the child is born. We'll talk about that later, which is actually the synapse and the axons, which occur more after birth as well. Again, the last week is more about gaining fat, which is just to help the child regulate temperature. And also the lungs for the exchange of air, which is another problem with a premature infant, is the inability to have the lungs operate properly, as is temperature control, and as is, of course, swallowing and digestion. So this all happens in the last month. Most of the weight gain occurs as well for a pregnant woman in month 8 and 9, because that's when the fetus is gaining the most weight for their layer of fat, right, to help them prepare for the outside world. Okay. Anything glaring here? We're all good? So here's the age of viability, and again, approximately 24 weeks of gestation. So here it is, and I mean, a lot better looking than this guy, right? So things are starting to become more proportionate. And at this point in time, if this child were born, it stands a pretty good chance of survival at 24 weeks. This is phenomenal when you think about it, because even in the late, even in the 60s, I had a brother who was born 11 months younger than me. And I was born in November. He was born the following September. And he was born premature. He was born at 8 months. 8 months. And this was in Newfoundland, and didn't have the technology that we have today. And this is only, we're talking 40 years ago. And his lungs weren't fully developed, which is normal for 8 months. You know that. That's when they start to develop. He didn't survive. 3 days was all, and they couldn't save him. And it's very sad, because had that happened today, that would be a non-event. 8 months old is a non-event. The incubators and all the equipment that they have today. So just in that, that little bit of time frame, we've come great leaps. I mean, 24 weeks, you can survive. We're in this poor child 8 months, right? So again, in that little time frame, we've learned a lot in the prenatal areas of the hospitals.